In the prior lecture that I suppose this year you guys saw by recording of me, it was an overview lecture. It's an overview lecture for this quarter, quarter 3, which is broken down by the major components of the central dogma. Today, we'll begin with the process of DNA replication. We're going to get as far as we can with the time that we have. Finish it up next week when I get back, and then we move on to the other components that were given in that overview lecture: transcription, translation, and then there's a relatively new lecture, which we call extra dogma, which will deal with things outside of DNA makes RNA, makes protein. We're going to talk about replication in not in the context of a disease. As you might imagine, if cells can't replicate their DNA, there's not a disease that results from that. It's called no life. And so instead, we're going to talk about the man-made ills of chemical and biological warfare that have mechanisms of action related to their effects on DNA replication. Humans have a long history of exploiting chemistry and biology as weapons. If we're talking about the pitch and sulfur of the Spartans, or arsenic by the Chinese, or the biowarfare of using Yersinia pestis by the Mongols. We're going to talk about two weapons in particular, one that's chemical and one that's biological. The chemical weapon is called mustard gas. The full chemical name is shown on the first bullet point there. As a chemical compound, with respect to the way in which it intersects with biology, is that it acts as what's called an alkylating agent. Alkylating agents add alkyl chains to whatever they react with. In the case of mustard gas, these alkyl groups here react with nitrogens. There are many nitrogens in biological molecules, proteins, and DNA. And the reason why this chemical compound acts as an alkylating agent are those chloride groups at the end. For those that may be taking organic chemistry or aspire to take it in the future, the chloride's a good leaving group, and so the nitrogen comes in and attacks there, and then you have a covalent cross-link, covalent modification with that alkylating agent. It was synthesized originally over 200 years ago, and the biological consequences of mustard gas exposure are extensive. The first one to be aware of is that it is a vesicant. Blue term, vesicant are agents that cause blistering. Ugly picture coming up on the next slide. And the reason why it was used extensively in World War I before chemical weapons were outlawed internationally is that, as you can see by the chemical structure, pretty easy to synthesize. I don't know if you do it in organic chemistry lab, but it looks like something that would be possible. Pretty easy to disperse, stable over long periods of time, and really hard to deal with once an exposure has occurred. As I alluded to, symptoms are really nasty. All right, erythema, this is redness of the skin or the mucous membrane. It affects mucous membranes really extensively. Conjunctivitis, hand in hand of that of a mucous membrane. The inflammation on the eyes and the eyelids, upper right. Its mechanism as a vesicant, it causes blistering, burning on the skin. We have mucous membranes in our nasal passages. Smells, apparently, like mustard or horseradish, and this is where it gets its name. And all of the airways will become inflamed and have these adverse reactions. So the throat, airways, proceed to vomiting, and the statistics on how it acts as a chemical agent, LD50, that is the lethal dose that kills 50% of the people or organisms that are exposed to it, is on the order of 1.5 grams. And if a handful of milligrams make it into the lungs, because you have a lot of surface area there that's all mucous membrane, it's much smaller than that. I mean, this blistering affects, as you can see, it's as a function of surface area. So the volume of the individual relates to their surface area, and it's because of the blistering agent. The biowarfare agent we'll talk about is anthrax. We're going to talk about first the natural life and ordinary residence of the anthrax bacteria, Bacillus anthracis, and then we'll talk about how it can be weaponized. So Bacillus anthracis is ordinarily found in the soil. I think you've done other botulinum toxin, right? Some other bacteria over the preceding quarters. Also soil-dwelling bacterium. It's when those bacteria end up in environments that they don't ordinarily encounter that they can cause very severe disease. Bacillus anthracis likes to infect large vertebrates, such as sheep. That's why it's called wool sorter's disease. I'll show you the hand of a wool sorter in a moment. And another characteristic of this bacterium is that when conditions are not favorable for growth and proliferation, it will form spores. What are spores? Spores are highly dormant forms of an organism that are adverse to adverse environmental conditions. So if the water goes away or the humidity is too high, it gets really cold or really hot, there are organisms that can go into this sporulated form, and they weather those environmental conditions and wait for it to get better, and then they come out of the spore form and can replicate again. Anthrax is but one example of organisms that will go into this sporulated form. The sporulation characteristics of Bacillus anthracis are one of the things that is attractive for weapons makers. Mustard gas is stable. Anthrax spores, likewise, very stable. Not too hard to grow. The spores themselves with the right carriers can be dispersed. The mortality, I'll give you some numbers on that, is really high. And it's a one and done infection. It's not like, it's not a propagating infection, like releasing some sort of virus or things that would be difficult to contain. So here, the reason why it has been used in the past is because you can target it to a particular location, and then it doesn't go any further than there. Before any weaponization of anthrax, the common way for humans to pick up an anthrax infection is on the skin. Cutaneous anthrax, and this is where the wool sorter's disease come from. So somebody's shearing a sheep, and they nick themselves with the shear, and their hands are all in the sheep wool, and that large vertebrate is infected with anthrax, goes onto the skin. That infection is very local. The symptoms come up as one of these necrotic lesions on the skin here. There'll be some local inflammation, lymph nodes, these things that involve trafficking of immune cells. We've already gone out of the cellular immunology quarter, so secondary lymphoid organs and things, those will be inflamed. The necrosis, earlier quarters we talked about apoptosis, programmed cell death, cell suicide. That might have been Dan, one of the early lectures. That is an instructive cue for cells to die by their own internal mechanisms. Cell necrosis is sometimes called cell murder. If apoptosis is cell suicide, necrosis is cell murder. That is where some sort of noxious agent attacks the cell. Could be a chemical compound, could be a pathogen such as anthrax, and causes the cell to die in a much less regulated or unregulated form. And this is what occurs with the cutaneous anthrax infection. The course for weaponized anthrax via inhalation to the lung proceeds via an entirely different sequence of events. One has to aerosolize the spores. They are inhaled. The infection thus begins in the lung. For about a week, nothing happens as they sit there in the lung. Not shown on the slide here, but what occurs is they get taken up by macrophages in the lung. You heard some about macrophages in the last quarter. And it's inside, they get phagocytosed. They're particles as far as the macrophage is concerned, phagocytes. So phagocytose the anthrax spores. And as a result of getting phagocytosed by the macrophages, that creates environments that are more favorable for the anthrax. It then goes out of the sporulated form, begins to replicate, and now you have a rampant infection in the lung. Within a couple of days of symptoms, mortality is guaranteed unless treated by only a handful of antimicrobials have been shown to be effective for this bacterium, one of which, Cipro there, we'll talk about in the next class. LD is not really an LD50 if everybody dies, but it's on the order of tens of thousands of spores. And just a couple of symptoms, we say for completeness. Symptoms looking like an upper respiratory infection. And then the bad things near the end, profuse sweating, for the pre-meds in the audience are diaphoresis, muscle pain, myalgia, and then septic shock in the end stages of disease. This is a combination of two things, shock, low blood pressure. It's a systemic condition. And where the sepsis and the septic part is coming from is as a result of this overwhelming infection caused by the bacterium. It starts in the lung, propagates there, eventually goes into the bloodstream, and then the individual goes septic. Why on earth? These seem like strange bedfellows, right? Why would you bring these two together in a lecture on DNA replication, no less? The answer is, as you will see, mustard gas, it's one of its mechanisms of action is by performing chemistry on our DNA. And then as a consequence of the chemistry performed on the DNA, that has adverse impacts on DNA replication. Reciprocally, that antimicrobial Cipro that I just mentioned very briefly on the preceding slide is a synthetic antibacterial compound whose mechanism of action is to disrupt specifically bacterial replication. And so it's exploiting differences between the way that we replicate our DNA and the way that Bacillus anthracis replicates its DNA. So we have a therapeutic angle to this lecture, and also a real nasty World War I type of angle to it. Let's start with a quick review of a slide that was on the pre-lecture material from the earlier slide. This is going to involve a lot on DNA. We should remind ourselves what DNA is made up of and what it's about. Double-strand helix. The two strands are anti-parallel to one another. There's a polarity with every chain of DNA determined by the positions of the 5' and 3' ends of the ribose sugars. And so depending upon how the connections here are made, 3' to the 5' carbons of the ribose sugar, that determines the 3' end of the DNA versus the 5' end. The information contained in the DNA is located not in the phosphate backbone and the ribose, deoxyribose, but instead in these nitrogenous bases in between. These nitrogenous bases, the A's, T's, G's, and C's, pair up with one another via hydrogen bonding. I talked about hydrogen bonding at the beginning of the first quarter. Hydrogen bonds are shown by the little dashed arrows on the picture on the right. And the base pairing is very specific. A is base pair with T's and via 2 hydrogen bonds, and G's base pair with C's via 3 hydrogen bonds. Because of the specificity of the hydrogen bonding, a proper base paired set of double-stranded DNA, one knows the information on the other strand if given the sequence on the complementary strand. So that back and forth of the hydrogen bonding of the A's and T's and G's and C's make them what's called complementary to one another. I already talked about the anti-parallel nature. I already talked about the base pairing and hydrogen bonding. These all come to the fore in the setting of DNA replication because the enzyme that catalyzes the doubling of our DNA and every DNA on Earth is an enzyme called the DNA polymerase. As the name implies, it is an enzyme that lowers the activation energy barrier for the polymerization of nucleic acids into a DNA strand. And DNA polymerase catalyzes that reaction in a specific way by exploiting that complementarity. if one takes away a strand of DNA, and one knows the rules of how to base pair A's and T's and C's and G's, it's possible to then regenerate another strand of DNA based on the information in that single strand that was used as a template. All right, so it's the beginning rules to synthesize the new daughter strand. For DNA replication to work effectively, meaning for all of that magical stuff that I just described in the cartoons from before, there are several challenges that DNA polymerase or the DNA replication machinery in cells need to overcome. And the upshot is that DNA polymerase itself can't do the entire process of DNA replication. It's going to need help because of some limitations in what this enzyme can do. It's an amazing enzyme, but it can't do everything related to DNA replication. First limitation of DNA polymerase is that it cannot separate or melt the double-stranded DNA that would, where one would like to use one of those strands as a template for replication. In the slide before, I just whited out the one strand, but in reality, there's an energetic barrier to separating the hydrogen bonds that keep the DNA as the double strand so that there's enough space for the DNA polymerase to get in and elongate and create a copy. Problem number 2, I think that this was said in the overview lecture, DNA polymerases can only elongate a strand that has been started. If there's a little piece of nucleic acid, double-stranded nucleic acids hybridized together, complementary, DNA polymerase is off to the races and can keep running and can fill in the longer piece if there's a short way to start it. But it can't start de novo. And then the third challenge is if you put together what I said in the preceding slide about the anti-parallel nature of the DNA, and then the additional characteristic of DNA polymerases in that they catalyze the elongation of DNA by synthesizing new DNA from the 5' to 3' direction. This is the only direction that they can synthesize DNA. This is the reason why, I keep harkening back to the one lecture I've given you up to this point. Remember in proteins, N-terminus to C-terminus? DNA is 5' to 3' all the time, never the other way around. And the reason why is because DNA polymerase can only make it 5' to 3'. So why should we read any differently than DNA polymerase? But if DNA polymerase can only replicate from 5' to 3', and as I said, the parent strand is a double strand that's anti-parallel, the directionality of DNA polymerase is going to require polymerases moving in the opposite directions depending upon which strand is operating on. We'll see if we get that far today, but if not, then next week. What we'll do now is we're going to deconstruct those challenges and some other ones that I didn't lay out explicitly to DNA replication, and then we'll try to put it all together at the end to see how it's a real coordinated effort by multiple biomolecules in the cell, one of the major ones being DNA polymerase, but several others critically involved. The first question, which relates to the challenge that I said in just a moment ago, is how do you get this whole thing started? Irrespective of the organism, the genome is going to be double-stranded, as long as you're not talking about a virus. Right? Double-stranded, and it will be stably complementary and base paired with itself. When replication is triggered to occur, at some point that the whole replication machinery needs to start somewhere, and then all of the sequelae that I'll talk about in a moment need to occur. So how does it begin? It begins at this location in a genome called a replication origin. Replication origins is where DNA replication originates. That's a pretty easy blue term. And one of the characteristics of the replication origins is that they are recognized by origin-binding proteins. I'm laughing because this sounds so ridiculous right now. It's like I define one thing and I tell you something else meaningless that defines what that thing was. This is the field. We'll say more about what these origin-binding proteins are, but suffice to say now at a high level, these act in many ways as a nucleating event to set off all of the other steps for DNA replication. So those origin-binding proteins look at particular characteristics in the DNA and identify them as good starting points for DNA replication. In prokaryotes, the characteristic of origins of replication can be assigned to the DNA sequence that comprises that origin of replication, and that those shared characteristics of the DNA in origins is called a consensus sequence. I want to clarify that the term consensus sequence does not apply uniquely to origins of replication, but it's any type of shared sequence characteristic that defines a shared function biologically. In this setting, we have a consensus sequence for an origin of replication. All the A's, C's, G's, and T's down below are not blue for a reason. Maybe one thing that I'll clarify, because it's come up in years past for the shorthand, N means any nucleic acid, A, C, G, or T, meaning it doesn't matter what the identity is of it. In prokaryotes, bacteria, we can identify these types of consensus sequences like shown in the cartoon. In eukaryotes, the origins of replication are not so cleanly defined. We can't define consensus sequences like this. Yet, nevertheless, there are still origins of replication. There are reliable places where DNA replication initiates along our chromosomes. It's just we can't figure out a consensus sequence for them. They usually have an enriched stretch of AT base pairing. First question to the group, why might that make sense thermodynamically for an origin of replication to occur in an AT-rich region? You see it also in the consensus sequences up there. Sure, yeah, and so the rationale thermodynamically is that the AT base pairing has one fewer base hydrogen bond holding it together compared to GC base pairs. So it should be easier to melt regions that are AT-rich. I want to speak briefly on how these consensus sequences are identified. And this is done through an area which you've probably heard of, but you may not really know what it's all about, called bioinformatics. We've sequenced a lot. And so we know a lot of bacteria. We know a lot of prokaryotes. We know a lot of DNA sequence, and we know a lot of these bacteria initiate their DNA replication, right down to the bases. And so what bioinformaticians can do is align those DNA sequences that are brought together by the fact that they're all origins of replication. And these alignment types of approaches are very similar to the amino acid alignments that I talked about in lecture 1. And then after they've been aligned as optimally as possible, one can look for sequences, stretches of bases that are shared by each one of those origins of replication. We can highlight them yellow, and we can call them a consensus sequence. This is the gist of how these consensus sequences are defined. One wrinkle to this lecture, when I used to give it a couple of years ago, I would only focus on prokaryotic DNA replication because it was somewhat simpler. But in the last year or two, I've modernized it to do a compare and contrast between prokaryotic DNA replication and eukaryotic DNA replication. I finally found a satisfactory review for me that I could wrap my head around and gave enough detail that I could relay to you about the similarities and the differences between the ways that prokaryotes and eukaryotes replicate their DNA. The reason why I want to present it to you is because it's really fascinating. They're very, very different organisms, one might think that something like DNA replication would be deeply, deeply conserved. Not so true. It's sort of like flight. So you think it'd be hard to fly, but flight's been invented 3 times: bats, birds, and insects. So similar type of analogy here for DNA replication. Some of the themes are similar, but a lot of the details differ, and I'm going to try to do a compare and contrast when we talk about it. This slide, left-hand side, prokaryotic DNA replication. Prokaryotes, circular genome, one big circle. Us, and other eukaryotes, chromosomes, linear genome, several of them. Right. When the DNA replication machinery becomes activated, the first thing that happens for both of these is you need to start somewhere. So all of us have origins of replication. Bacteria have one and only one origin of replication all the way around their DNA genome, their circular genome. In contrast, we have multiple origins peppered all along those chromosomes, and by extension, all of the chromosomes have multiple origins of replication. But the idea of initiation is the same. Once the replication initiates, the strands separate, and replication of those parent strands to give rise to daughter strands begins to occur. When we pull apart the 2 parent strands and begin replicating, for the reasons that I described about the antiparallel nature of the complementary base pairs, replication happens in both directions in a symmetrical fashion. I'm trying to dodge some of the words that you're going to learn next slide. But the same thing is happening almost like a mirror image to one another on one side of where the melting has occurred as on the other side. Those separated parent strands, just one of these here, is called a replication fork. Hopefully that's self-explanatory. Fork. All that's pretty much the same in eukaryotes. Maybe a little bit of the terminology changed. Oh, I'm getting ahead of myself. Okay, replication fork. I already talked about parent and daughter strands. So the parent strands are the ones that are going to act as the template for DNA replication. The daughter strands are the newly synthesized DNA that has been elongated off of those parent strands. Because we have chromosomes that are not circular and you have multiple origins per chromosome, when these replication events initiate, you get something called a replication bubble. To be honest, it's comprised of 2 forks. They've got 2 forks on either end. The only distinction is that you have them all across the chromosome rather than in one location for a prokaryotic genome. And the last thing I'd like to reiterate is over here, the origin has a consensus sequence defined to it. We can make bacterial-like circular pieces of DNA and put in one of those bacterial origins and have replication occur in bacteria. A lot harder to do that with a eukaryotic origin of replication. The last thing that I want to say here, because we have these multiple origins, they're going to coalesce eventually as they nucleate, expand, grow. When they fuse together, now those individual daughter strands will get joined together through a process that I'll talk about in a moment, and now we have fully replicated linear chromosome sequences. Do we have questions up to this point? Let's dive in then. We'll begin with this process of initiation. As I said, DNA polymerase cannot melt apart the strands. There are proteins that can. They're called initiator proteins. And what they do, that's an example of an origin binding protein. It recognizes the origin under conditions for DNA replication, binds there, and depending upon the specifics of the initiator protein, it can either bind and separate. Is there a debate I can clarify? There is only one origin of replication in prokaryotes. There are multiple origins of replication in eukaryotes. These initiator proteins, depending on if you're a prokaryotic or a eukaryotic organism, beyond that similarity, differ in the details. I talked about protein structures, tertiary structures, all those things. Most of the cartoons that I showed you before were surface maps. Remember, those for hemoglobin, all that other stuff. We also showed ribbon diagrams, alpha helices, beta sheets. Sorry, I like to bring back all that stuff you think you never needed to know again. I'm going to do that a lot. That's why I like teaching the second half of this class. These are protein structures also. It's a different display. The circular barrels, it's a barrel in ribbon diagram. The barrels are alpha helices. So graft on that helical look when you're considering the structure of these proteins. On the left-hand side is the initiator protein for prokaryotes. It's called the DNA A. DNA A binds to origins of replication and will separate the strands. In eukaryotes, we have a complex, an origin of replication complex, comprised of 6 proteins, each one called ORC. So we have ORC, 1, 2, 3, 4, 5, 6. The color coded there, so it's a hexameric assembly. And that hexameric assembly, you see the hole in the middle? That's where the DNA goes. All right, so it assembles around the double-stranded DNA at the origin of replication. ORC will not separate the DNA, but what the purpose that it's serving is as a docking site. It's like landing lights for the enzymes that will separate the DNA. DNA A and ORC have that ability to navigate to, recognize, and bind to these origins of replication. What happens next? Irrespective of whether or not you're DNA A and you can partially open the DNA, or you're ORC and you just land there, there needs to be a dedicated enzyme that separates the strands once that fork starts moving. Because the ORC or the DNA A will just initiate the event. It doesn't move beyond that. The dedicated enzymes that will go and unwind the fork need to have enzymatic activity, need to expend energy, and the enzymes that do that are called helicases. Helix, unwinding the helix. That's the catalytic reaction that they're enabling, that melting of the duplex DNA. Helicases hydrolyze ATP. This is where they get the delta G to enable that to be thermodynamically favorable. Here we have another similarity and distinction between prokaryotes and eukaryotes. Prokaryotes, their helicase is called DNA B. So DNA A to DNA B. You can see why we used to teach it in prokaryotes. It's pretty easy, right? Eukaryotes have another complex called the MCM complex, MCM 2 through 7. MCM stands for mini chromosome maintenance. It relates to the historical origins of how these proteins were identified. This MCM complex and some accessory proteins here that are here because this is where I got the image, but I'm not going to talk about them any further, will self-assemble around they interact with ORC, or DNA B interacts with DNA A, and then we'll unwind those DNA strands. The nature of the unwinding, though, differs pretty substantially between prokaryotic and eukaryotic helicases. DNA B, its self-assembly, you can see another hexameric protein, so there's 6 subunits of DNA B that come together around the single strand. It binds to the template strand and moves along there as it does its unwinding. In the MCM complex, you have the same, and by virtue of that, it's moving in the 5' to 3' direction. As it's hydrolyzing ATP, unwinding the DNA, it's moving in a polar direction on one of the strands. The MCM complex is doing the same thing, but is doing it on the other strand. So its processivity, the motion that is occurring 3' to 5' instead of 5' to 3'. Still another hexameric assembly, different, no similarities in the protein sequence. There's maybe some structural similarities, but what they're doing on the DNA is rather different. Yet functionally, the effect is the same, nothing in DNA. Once helicase has separated the DNA strands, if all it did was separate the DNA strands and just keep moving on, what will occur is that those two separated daughter strands, the two separated parent strands, because right now they haven't been replicated, will just anneal right behind the helicase. They're right next to one another. Hydrogen bonding is still occurs. What keeps those single-stranded DNA templates apart are a class of proteins called SSBs. SSBs, single-stranded binding proteins. And what they recognize are single-stranded DNA, coat them with the protein, that's what they bind. And as a result of the protein binding to the single-stranded DNA, they block that hydrogen bonding that would otherwise enable the two parent strands to reanneal, coalesce, bind. And then the final accessory protein, but instrumental protein in DNA replication, is another category of enzymes called gyrases. Gyrases are probably the most difficult to get one's head around topologically. So I'm going to give you a demonstration in just a moment so you can have that stick in your mind. But the upshot is that DNA gyrases are another ATPase. They use energy to relieve the thermodynamic hurdles of taking a double-stranded helix and pulling apart the strands. As you pull apart the strands, you get supercoiling, that was said in one of the pre-lectures, overwinding of the DNA on either side of that replication fork, or upstream of either one of the replication forks or the replication bubble. So first, before I do my demo, I want to talk a little bit more about the gyrases and how they fall into a broader class of DNA chemically modifying enzymes called topoisomerases. What topoisomerases refer to, so gyrase is a type of topoisomerase, although there are other types of topoisomerases. Where the topoisomerization relates to is the coiling and the winding of DNA. So any enzymes that change the coiling content, the winding, overwinding, or underwinding, would be considered topoisomerases. The first type is called type 1, and it acts very simply if there is a piece of double-stranded DNA that has gotten tangled on top of one another. This could occur via the chromosome condensation that occurs during mitosis. So you have open chromatin, the whole thing collapses on top of itself to do chromosome segregation. That can create some tangles that need to be resolved. You can also have some tangling that may occur during transcription. So there could be some hurdles there that need to be overcome. And type 1 topoisomerases will resolve these tangles. And so with the tangling, what it will do is snip one of the DNA strands and then loop it around to relieve the supercoiling, and then bring those two cut strands back together again. End of the catalytic cycle. So cut, unwind, rejoin again, and that's what the enzyme does. Because an overwound or tangled piece of DNA has some energy associated with it, that energy associated with it is actually exploited by the type 1 topoisomerase to cut, unwind, and then rejoin. Therefore, type 1 topoisomerases don't require any energy expenditure to be able to do that catalysis. They use the energy of the coiling to accomplish it. And if I haven't already said it 2 times, I will reiterate. I give this to you because gyrases, which are involved in DNA replication, are a topoisomerase, but they're not type 1s. So type 1s DNA condensation, transcription, but not DNA replication. And here's a picture of what topoisomerase looks like. Type 2s also achieve unwinding, relieving supercoiling of DNA, but the geometry of the trick that they pull off is somewhat different. And gyrase is a type 2 topoisomerase. Here, you can have the supercoiling occur to such an extent that the double-stranded helix will almost like be wrapped up on top of itself. And what type 2 topoisomerases do is they'll cut, rather than one strand, cut both strands of one of those tangled up pieces of DNA, and then pass the cut double-strand DNA through the one that has been uncut, and then religate it on the other side of the cut. That's kind of confusing, and the cartoon doesn't help very much. This I'll show you with my demonstration in a moment. In contrast to the type 1s, which are one cut, this is a double cut and then a double joining at the end. Here, one does need ATP hydrolysis to get enough free energy to be able to do the double cut, pass through, and paste it together on the other side. And I will repeat that DNA gyrase is a type 2 topoisomerase. Here's an alternative cartoon of what it looks like. The green is in the DNA. The green is the double-strand DNA that's being cut. Blue is your type 2 topoisomerase. Once it's open, we have now another double-stranded DNA, orthogonal, perpendicular to the one that just got cut. It passes through the space created by the cutting of the two strands that were broken apart by the type 2 topoisomerase. Whoops. Oh, there's not another animation. Then it comes back together. It's demo time. 1, 2, 3, 4, 5, 6, 7. Can you come up? Yeah, this is good. If I can keep, we'll start with you two, and then I'm going to keep you all in a reserve as I need you. You're going to be one side of the DNA. You're going to be the other side of the DNA, so if you'll come over on the side here. Double strand. Double strand. Let's get that helix going. This could be a linear chromosome. It could be a flat enough view of a circular prokaryotic genome. Same thing's happening. Keep spinning. Next up. All right, you're going to be our initiator. Come underneath. And pick a site. I'll put your label on first. Look. Find an origin of replication. Actually, do me a favor. Can you do it on the right-hand side? Because I don't want to have to go back to Home Depot and buy more rope. All right? And then bind there. OK. Now, I'm going to peek over your shoulder and not try to stress you out now. So if you are ORC, what should you be doing? Mm-hmm. Trick question. Yeah, just hang out there. Like nothing. Nothing except for something like this. All right. If you're DNA A, what you were doing before. Yeah, just a little bit. Yeah, a little bit, open it up. Yeah, just a little bit of an opening. Okay. And can we move the DNA a little bit forward? Because I want to, there we go, that's good. All right, we're going to have two helicases. that will come in, and we'll start it with the DNA A. One helicase needs to be on the other side of the DNA A. And then the other helicase will come in here. Now you've separated the strands just a little bit, and now each of those helicases, start moving them apart. That way, good. And now I'll slow it down for a little bit. I'll need some single strand binding proteins over here. As you move that way, keep going. Try not to give yourself a rope burn. You can use multiple hands to avoid the rope burn. But the single strand binding proteins, there. Okay, now the initiator protein, are no longer needed. Okay, come out. Keep going. I'm going to come over here. Keep going. I get to be gyrase. Keep going. This one, let's go helicase. No, whoa. We might be doing it on the other side then. Okay, have you held up over there? All right, keep going. Just hold it tight. Keep going. Yeah, like really, 2 hands, double hand action. You're not going to break it. Okay. Now, okay, stop for a second. Now don't let go, but just, okay, there's the supercoiling. All right, so you see how unwinding? This is the energetics that need to be relieved because it's getting hard to unwind, right? Same thing for the enzyme. Let's hypothesize that I'm, first just by way of introduction, let's pretend I'm a type 1 topoisomerase. Don't let go. Let's see how I do this. I'm in rope preservation mode. One cut, and I can unwind. This one's pretty obvious, right? I'm relieving, at least locally. Oh, did I not cut that one all the way through? Good enough. You get the idea. All right, so it's unwinding in this way. If I'm a proper gyrase, make sure I don't snap my fingers off here. You hold this end for me. Be part of the gyrase. This now, I'll need you, the equivalent of going like this. This way. I'm going to do this a couple times so you can see the parlor trick. No, keep it there. Okay. Yeah. If I do it in the right direction, I'm doing it in the wrong direction. Sort of. Maybe try. Maybe I shouldn't. Let me see if I go. Put your finger out from there. Yeah, you keep it. Hold on. As I do this, can you see that it's unwinding? All right. A lot looser than it was before. This is exactly what DNA gyrase is doing, multiple times over as it's in front of those replication forks going forward. All right, let's give our helpers a round of applause. Thanks. The demonstration illustrated where gyrase comes into play immediately at the replication fork, as you saw on both sides. Type 2 topoisomerases are also important for circular genomes, prokaryotic genomes. At the end of that replication event, all the way through, when you have the parent genome and the daughter genome, they will be interlinked together with one another like this, and you need another type 2 topoisomerase to cut, pass through, and rejoin again to separate the two. This is an electron micrograph image on the right-hand side showing what we're talking about. There's a question in the back.
So type 2 topoisomerases are so much more effective in discontinuing the supercoil. Why would the body use type 1?
Yes, and so the question is, if the gyrases and other types of type 2 isomerases are so magical, why would there be any role or need or evolutionary conservation of the type 1 topoisomerases using the mechanism that I described at the beginning? I have a couple answers to that. I'll give a handful of them. One, let's think from an energetic perspective. Each time the gyrase needs to do one of those looping, I was just doing it here, but in reality, the enzyme is consuming ATP each time it passes through. And as I described for the type 1 topoisomerases, that's energetically neutral. And so that's one advantage. The second, not always do you have a tangle that has another strand ready to pass through in the way that, like I described. In DNA replication, it's an intrinsic characteristic of the moving replication fork, but a condensed chromosome or a random tangle, there may just be a little nick there or a little bit too much supercoiling that you can't do that type of passing through. No magician hoops to do the trick with, and therefore having type 1 topoisomerases in and around to relieve those would be beneficial. We have initiated the replication fork. What comes next? Haven't said anything hardly except for all the limitations of DNA polymerase. We should say some more things about it. As I alluded to, DNA polymerases catalyze the growth of DNA polymer. They do that by adding nucleotides on one end of a piece of DNA or a piece of nucleic acid, and that end is the 3' end. And this is where the rule of growth occurring from 5' to 3' comes from. For DNA polymerase to do that catalytic addition of a nucleotide, it needs a template to be able to discern what is the appropriate nucleotide base to go in at that next position on the 3' end. And that base comes in the form of a high energy monomer called a deoxynucleotide triphosphate, or DNTP. And that DNTP, the deoxyribonucleoside, that has the base, ribose sugar, all that fun stuff. The triphosphate has the energy. And when the incorporation of one of those nucleotides occurs into a growing strand, so 5' to 3', what gets released, or the chemistry that occurs, is that this free hydroxyl attacks the alpha phosphate of the triphosphate portion of the nucleotide. And then what gets released are the beta and the gamma phosphates as an inorganic pyrophosphate. Inorganic because there's no carbon in the molecule. That hydrolysis event is highly thermodynamically favorable, and that's where the energy sources come from. The third thing that speaks to one of the limitations of DNA polymerase, that it requires to be able to do that elongation event, is that it needs some stretch of nucleic acid to elongate. It can't start the DNA synthesis process itself, but it can elongate an existing strand. And that piece of nucleic acid that starts the process off is called a primer. A word or two about primer and priming. People heard the phrase prime the pump? I'm really old now. Okay. Have you ever painted anything and had to put on primer? Do you know why? Okay, I got something. I'm glad I had two here. What's the primer for? Great. So to be able to paint, you need to paint. Yes, that's why it's called a primer. And I won't even tell you about priming a pump. If you've never used a water well before, it's not helpful. So this is the notion of the priming. It's basically you need to have something, you need to have the thing for the thing to occur. All right? So it's the same thing. You need to have nucleic acid there to be able to make more nucleic acid. That's why it's called a primer. But the primer is not laid down by DNA polymerase because, as I said, it can't create that de novo. Instead, there's another enzyme called primase. What do you think that it does? It is another category of enzymes, not a DNA polymerase. It is in a category of enzymes called a DNA-dependent RNA polymerase. And what it does is take that DNA template and lay down complementary RNA instead of DNA. RNA polymerases, like future RNA polymerases that you'll hear in the forthcoming lectures, they can initiate nucleic acid synthesis wherever, as long as they have a template. That's how genes get transcribed. And so what primase is doing is using a similar principle, recognize the DNA, and lay down a short stretch of RNA primer, and then DNA polymerase will recognize that RNA primer and begin to elongate. So they work together. And that priming ability, to emphasize, is because we're talking about an RNA polymerase, not a DNA polymerase. The major elongating polymerase, a DNA polymerase, in prokaryotes is called DNA polymerase 3, and in eukaryotes it's called polymerase delta, or Pol delta, Pol 3, Pol delta. These are large multi-subunit proteins, all of them. The eukaryotic polymerase is bigger than this. They all share a couple of common functions. I've already harped multiple times on the polymerase activity, 5' to 3'. But these elongating polymerases have an additional enzymatic activity that is important for Pol 3, Pol delta, to elongate DNA faithfully, with fidelity, meaning accurately. If it makes errors, that's called mutations, that's called cancer, that's called all sorts of nasty things. So it's in all our best interest not to put in any errors when we replicate our DNA. And the enzymatic activity that dramatically reduces the error rate of our polymerases is this 3' to 5' exonuclease activity in DNA. It's encoded in one of the subunits of DNA polymerase. And this exonuclease activity is through this process called proofreading. And what proofreading entails is a check, if you will, of how an incorporated nucleotide looks with respect to the parent strand, the template strand, off which the DNA replication is occurring. We'll go through this cartoon. We have the template strand here, elongating daughter strand. The next one, the next nucleotide that should go in is an A. But the polymerase has incorporated a G. The G doesn't base pair appropriately with the T and creates a little offset there. It doesn't lock into the double helix the way that the appropriate base does. When that happens, there is a pause in the polymerase activity, and it doesn't elongate anymore. It doesn't stop entirely, but it slows down, and that slowing down gives enough time for the polymerase's 3' to 5' exonuclease activity to kick in and resect that inappropriately base paired and incorporated nucleotide, and then the enzyme can try again. And it's 3' to 5' because now you're cutting in the opposite direction. You're undoing, the enzyme is undoing what it just did. And it's called an exonuclease because what it's cutting, the nucleic acids that are being cleaved, that are being hydrolyzed, are from the end of that DNA. For completeness, if we have exonucleases, outside ends of DNA, we also have endonucleases, not relevant for DNA replication, but you'll see them for molecular biology, cloning, synthetic biology, use endonucleases all the time. They recognize the middle of a double strand and cut it in two. So that's where the endo and the exo comes from. And as a consequence of this base pairing, elongation, synthesis, and proofreading, what that DNA polymerase 3, DNA polymerase delta is doing is making the reverse complement, which means the complementary bases, but then in the opposite orientation because of the anti-parallel nature of DNA as it's doing a replication. Ribbon diagram, this is polymerase 3 interacting with the open strands of DNA here and then elongating. Talk about all these enzymatic activities of DNA polymerase. How can it catalyze multiple chemical reactions at the same time? Remember, I didn't teach you this, but I hope you still learned it. Remember enzyme kinetics? E plus S makes an enzyme substrate complex, giving rise to enzyme plus product. Polymerase, DNA with N-1 bases. Bound polymerase, DNA with N-1 bases. Polymerase, DNA with N bases now. If the enzyme is released, it's got to come back, find the template again, and start this whole process. This enzyme encounter substrate is usually the rate limiting step for most enzyme catalyzed reactions. It's finding and generating this complex. Once it's in that complex, the catalysis can happen pretty rapidly. This would be prohibitively slow for DNA replication to happen ordinarily. Therefore, one of the subunits of either both the eukaryotic and prokaryotic DNA polymerases is what's called a processivity clamp. These are subunits of DNA that of the DNA polymerase that wrap around that parent strand and the elongating daughter strand and keep the enzyme put right on where the elongation is occurring. The consequence of that is that rather than enzyme to substrate going to enzyme and product being released, it goes right back into another enzyme substrate complex because it never really released. And you just redefine what the substrate is. Now your substrate is DNA with N bases, and now you make N+1. And then N+1 goes and makes N+2. So this processivity clamp is essential for rapid elongation. All right, we have the third problem or challenge for DNA replication that I haven't mentioned. We'll see how far we get on this. And that relates to the mirror imaging that I talked about when you create a pair of replication forks. Initiate replication, unwind it, and then we have things occurring on each one of the replication forks. We zoomed in on one I was doing some scissor work. And all the cartoons here show one replication fork, not the other one. And they also focus on really one strand up to this point. Let's look at the two strands in context now. Things that I've mentioned already. Helicase, single-stranded binding proteins, gyrase, all that's good. Primase, laying down the RNA primer. DNA polymerase, using that RNA primer to begin elongation, and off it goes. In this cartoon, the bottom strand is just a cartoon. You could flip it around the other way just as easily. Right? The bottom strand, the newly synthesized daughter, runs left to right. 5' to 3'. You can see the teal ball polymerase moving, 5' to 3' and what it's synthesizing. This strand is called the leading strand because the DNA polymerase can just keep going with the replication fork as helicase and gyrase continue to unwind it and relieve the supercoiling. It can keep going until it's done. The opposite strand is anti-parallel, and in many ways it's like bizarro replication, right? Everything is happening backwards. Primase will still lay down an RNA primer on that other strand, but it is going in the opposite direction of the replication fork. And then when DNA polymerase binds and elongates, it continues to move away from the replication fork. And so therefore, as the replication fork is moving, there are multiple priming and elongation events that need to occur on that other strand, the so-called lagging strand, to be able to fill in all of the unreplicated DNA. Here's another cartoon of the exact same process, but you'll note that the leading and the lagging are on the opposite sides, and that's just because it's a different cartoon. But this is what a replication fork will look like as it's occurring, with one long leading strand and then a series of these smaller lagging strands being synthesized for the one that's going in the opposite direction of the growing fork. These little pieces are called Okazaki fragments. And this is a historic, who do you think discovered them, right? This guy Okazaki. And so they create a topological challenge that needs to be resolved when you want to get a complete picture of DNA replication. So the last piece that we need to figure out what to do with those lagging strands, those Okazaki fragments, and also what to do with that RNA primer, it ain't DNA. All right, we have to get all DNA at the end, is we need another polymerase called polymerase 1, Pol1, or Pol alpha, depending on prokaryotes versus eukaryotes. It has 5' to 3' polymerase activity. It has 3' to 5' exonuclease proofreading activity. All that stuff is the same. But the key additional enzymatic activity that this polymerase has is 5' to 3' exonuclease activity. We're moving bases in the direction of its processivity. And this is the key characteristic that will enable the damage bases, is if there's DNA damage and things. The key thing for DNA replication is that RNA primer. And for all of the things it does and what it needs to do, it's a bit slower as far as its catalytic activity and processivity compared to Pol 3 or Pol delta. But here's what's happening. This was the cartoon from the overview lecture. Parent strand, RNA primer laid down on what will become the daughter, Pol delta, right, Pol 3, elongation. There'll be another elongation event. Let's say that this is a lagging strand. All right, there's another elongation event. The Pol 3, Pol delta will come right up against the next RNA primer that it encounters. It has no 5' to 3' exonuclease activity, so it can't go any further. It will just stop, and then will eventually, the processimity clamp disassembles and it falls off. Then Pol 1, Pol alpha comes in at this junction here, and its 5' to 3' polymerase activity fills in the place where the RNA primer would go, while its 5' to 3' exonuclease activity is removing the RNA primer. So I think about it as like a plow and a cement truck on the backside. So it's plowing through that RNA primer, and then in the space that's being created, it's filling in the holes with deoxyribonucleic acid rather than a ribonucleic acid. And then the final step, so now we've cleared out the RNA primer, is the gap that arises after that removal of the RNA primer has occurred. So you've filled it in, but then there's still it can't jump over a place where the track is broken. Right? So the track is still broken at one place after DNA Pol 1 or Pol alpha fill in that RNA primer. This should be over here. That broken piece of track is repaired by a final enzymatic activity called a DNA ligase. Ligations, joining things together, tying things together. And what's getting ligated is the DNA. So a DNA ligase will fill that hole in the track. Now we have a contiguous stretch of DNA. We will put it all together. Two parent strands. We have initiator protein, recognizes an origin of replication, and may or may not begin separating the strands. where the major aspect of strand separation occurs is through the enzymatic activity of helicase, DNA B, or the MCM complex, pulling open those strands and moving the replication forks outward. That creates supercoiling on either sides of those replication forks that are relieved by the type 2 topoisomerase gyrase. Within the replication fork, we have priming RNA being laid down by primase that acts as the strand that is elongated by polymerase delta, or pol3, all the way up until it hits another RNA primer, which gets cleared out by polymerase 1, or pol alpha. And then the final gap in the track, so the final nick in the DNA, gets repaired by DNA ligase, completing the replication process. This seems like a good place to stop. Any questions? Yes? Are there multiple primers on the lagging strand? Yes, there are. Each one for each Okazaki fragment will be initiated with a primer, and then each one of those will have to get fixed by a pol1, pol alpha, and a ligase. Yes?
When polymerase 1 replaces the primer with the DNA, does it stop at the gap, or does it
The gap moves with the RNA primer. So it's elongating the strand that it binds to, plows out the RNA primer, and then when it stops there, the RNA primer pulling out, then it will unbind, and then the ligase comes in to fill the gap. Good luck on the exam. I will see you next week.